Greetings, I am Dr. Krishnathej. I received my PhD degree from Indian Institute of Space Science and Technology. My PhD work is related to development of electrochemical impedance sensor for early detection of cancer. My research areas include isolation of extracellular vesicles from serum, electrochemical impedance sensing, exploring organic materials for developing flexible random access memories. Polyethylene glycol, EDC-NHS coupling chemistry were used to isolate extracellular vesicles in various means, that is by using colloidal gold nanoparticles, silicon wafer coated with gold nanoparticles, screen printed electrode with gold working electrode. To synthesize gold nanoparticles, various input parameters were optimized and tested in UV visual absorbance spectrophotometer. The TEM results show that approximately 20 nanometer sized gold nanoparticles were synthesized. The synthesized gold nanoparticles are functionalized with polyethylene glycol and treated with EDC and NHS to make them suitable for conjugation with anti-CD63 antibody. These nanoparticles are then incubated with serum to isolate the extracellular vesicles. They can be centrifuged and pelleted at the bottom of the tube. TEM image confirms the isolation of extracellular vesicles. DLS and NTA were also used for characterizing the isolated vesicles. The benefits of working with colloidal gold nanoparticles is that they can isolate smallest sized exosomes. These small exosomes uptake is high in brain and by the liver, so it can be used for drug delivery to brain. And the most important thing is the isolation can be done by a simple benchtop centrifuge. Silicon wafer coated with gold nanoparticle can also be used for isolation of extracellular vesicles. The SEM image shows the silicon wafer coated with different volumes of gold nanoparticles. It was concluded from the NTA experiments that with the increase in the volume of gold nanoparticles used for coating the silicon wafer, we can significantly increase the number of extracellular vesicles that are isolated. The silicon wafer platform was also tested with the different sized gold nanoparticles and polyethylene glycol. And by increasing the size of the platform, we can increase the number of EVs that were isolated. The NDA results are presented here, and the isolated EVs were also tested by Western blotting with different surface proteins corresponding to the extracellular vesicles. The techniques established on silicon wafer platform were extended to screen printed electrode, and we found that approximately 10 power 8 extracellular vesicles can be isolated. Once the extracellular vesicles are immobilized on the screen printed electrode, a voltage is applied to the electrode for lysing the extracellular vesicles, making this technique suitable for microfluidic platforms to implement a detergent-free lysing technique. We optimized the electrical lysing technique by using sinusoidal and square wave signals with different amplitude and frequencies, and we found that approximately 50 to 750 millivolts will be suitable for lysing the EVs. After lysing, the extracted HER2 protein was quantified by using electrochemical impedance sensing. The electrochemical impedance technique established for HER2 protein was also extended for studying TSG101 protein. Both Faradaic and Non-Faradaic sensing are studied. At each and every stage of modification of screen printed electrode, we studied either charge transfer resistance or double layer capacitance. The results of the developed EAS system were cross-verified with the standard ELISA technique.